Good afternoon. Welcome to the Wednesday afternoon lecture series. My name is Alexandra Nita-Lazar. I am Chief of Functional Cellular Networks section at the LISB NIAD. Today I have this honor to introduce today's world speaker, Dr. Ileana Cristea, who is conducting what I think is some of the most exciting research today on cellular defense. But first I have a few announcements. The first concerns continuing medical education credits. The CME code for today is 37953. I repeat, 37953. Also, as you can tell, this is a hybrid lecture in which a few of us are here in the Lipset Amphitheater, but the vast majority of you are watching via NIH videocast. Those of you who are remote can still ask questions. You can do so by clicking on the button just below your videocast window that says Send Live Feedback. Click on that, type in your name and question, and we'll relay to Dr. Cristea at the end of her lecture. You can submit a question anytime. Please note that today's lecture is the final on this year's World season. We will take a 2-month break and begin a new season on September 7th with a lecture by Dr. Jill Banfield of UC Berkeley on the topic of microbial disease. Okay, our speaker today is Ileana Cristea, the Henry L. Hillman Professor in Molecular Biology at Princeton University. Her research lies at the interface between virology and proteomics. Ileana studies cellular mechanisms used in defense against viruses, as well as mechanisms used by viruses to inhibit or hijack host cell processes. Her lab employs multidisciplinary approaches, integrating molecular virology, microscopy, mass spectrometry-based proteomics, and bioinformatics. In recent years, Ileana has developed methods for studying spatial and temporal virus-host protein interactions, bridging developments in mass spectrometry to important findings in virology. For example, her laboratory has contributed to the emergence of the research field of nuclear DNA sensing in immune response to uncovering mechanisms underlying organelle remodeling and organelle structure-function relationships during infections. And they have discovered sirtuins as broad-spectrum antiviral factors. Ileana received her master's degree in medical chemistry, followed by a Ph.D. from the University of Manchester Institute of Science and Technology, under the tutelage of Simon Gaskell. She then pursued postdoctoral work in the mass spectrometry laboratory of Brian Chait at the Rockefeller University. Ileana joined Princeton University in 2008 as an assistant professor, became an associate professor in 2013, and attained her current position in 2016. Ileana is very active in the proteomics community. She is the past president of the American Human Proteome Organization, US HUPO, the past chair of the Biology Disease-Driven Human Proteome Project, BDHPP, of HUPO, and the chair of the infectious disease team of HUPO BDHPP. She has taught the summer proteomics course at Cold Spring Harbor Laboratory for more than 10 years and is senior editor for M Systems, associate editor for the Journal of Proteome Research, and on the editorial boards of Molecular Systems Biology and Molecular and Cellular Proteomics. She is also the recipient of many honors and awards, which include the Bordoli Prize from the British Mass Spectrometry Society, the NIH NIDA Avant-Garde Award for HIV/AIDS research, the Human Frontiers Science Program Young Investigator Award, the Early Career Award in Mass Spectrometry from ACS, the Mallinckrodt Scholar Award, and most proudly, the Princeton University Graduate Mentoring Award. The title of her talk today is Dynamic Organelle Remodeling, Viral Mechanisms to Subvert Host Cell Biology. Ileana, welcome to the NIH, and thank you so much for being with us today.
Thank you so much. Thank you for that very kind introduction, and thank you for the invitation. It's really a great pleasure to be here. I thank Alexandra Nita-Lazar, and I thank the organizers. And thank you for being in person, and also I thank those online for joining in today. I have to say it's so great to be in person for a seminar and see people in 3D. It's a huge difference, so, and it's a great honor to be here. So as you have heard, my lab is at Princeton University at the Department of Molecular Biology, where we integrate virology with proteomics to study at the molecular level infection with human viruses. And for today, I selected to tell you about some of our scientific adventures during the pandemic that have brought us to become quite interested and really fascinated with this concept of dynamic organelle remodeling that happens during the replication cycle of viral infections. So I'll tell you a little bit more about that and try to put this also in broader context. But first, I'll start with a couple of disclosures. I'm a co-founder and member of the Scientific Advisory Board of Everest Bio, a company that makes antiviral treatments by targeting sirtuins, and I'm also a member of the Scientific Advisory Board of 908 Devices, a mass spectrometry-focused company. What I will tell you today is not connected to my duties with these two companies, but just to let you know. All right, so just to start with a little bit of broader context, the replication cycle of a virus within a host cell is absolutely fully dependent on the ability of that virus to actually remodel organelles. So in other words, the changes that can occur in organelle shape and composition and function are absolutely essential for the viral infections, for the viral replication cycles. And one important illustration of this is given by infection with human cytomegalovirus. This is a beta herpes virus that is widely spread. It has a large genome and also quite a lengthy replication cycle of 4 to 5 days. So this virus has both the genetic capacity as well as the time to induce quite striking changes in the subcellular or in the subcellular environment during its replication cycle. So for example, for its entry, it remodels the plasma membrane, it remodels cytoskeleton and endosomes, and then following its nuclear replication, the virus also disrupts the nuclear periphery in order to allow the viral capsid to egress from the nucleus. Later in infection, the virus makes a new assembly, a subcellular compartment formed of Golgi membrane and endosomes here on the left-hand side, I show you this remodeling of the nuclear periphery that I mentioned earlier. So the assembly compartment that makes new viral particles that contains endosomes in them and Golgi membranes that are restructured. And later, for egress, also it hijacks trafficking processes, host trafficking processes. Another hallmark of HCMV infection is quite a profound mitochondrial fragmentation, and I will tell you a little bit more about this later in this talk. However, what's not fully understood is how these remodeling events are connected to some of the viral pathologies induced by this virus. So for example, cardiac disease or the oncomodulatory properties of HCMV. However, what's important to note is that organelle remodeling is essential for all viruses. So regardless of different viral genome sizes, different replication strategies, different cell tropisms, all viruses are united in their need for regulation of organelle function, organelle composition, and shape during the replication cycle. So for example, HSV1, an alpha herpes virus, also modulates the nuclear periphery for its nuclear egress of the viral capsid. Influenza A also induces mitochondrial fragmentation. The beta coronavirus, OC43, just like other beta coronaviruses, induces this remodeling of the ER. It makes these double membrane vesicles that are critical for the replication of this virus. So there are all these dynamic remodeling events that are occurring during infection. And this is something that we have focused on in recent years. So with the question of how and why, rather which of these processes are for the benefit of the virus or the host. And in order to understand this infection-induced rewiring of host cells, we have a multitude of methods available from proteomics, from metabolomics, from lipidomics. And today I'm going to try to also highlight some of the strategies that we have used over the years, and in particular in more recent years, and how we have learned about this intracellular reorganization from these different methods. So We have started initially by focusing on understanding the virus-host protein interactions that are driving some of these signaling cascades and some of these remodeling events in the cell. And of course, we have so many different approaches that we can use for studying protein interactions during the progression of an infection. We can use affinity purification, proximity labeling, cross-linking, and many protein microarrays. And these methods are providing us with the opportunity to either study interactions from a local, in a local perspective focused on one protein of interest at a time, or from a global perspective. And given our interest in organelle remodeling, more and more in these recent years, we became fascinated by the idea of trying to capture in one go what happens in the cell during infection and how these events could be coordinated. So one method that I want to highlight today is this thermal coaggregation profiling. And just to take you a little bit through the approach of this method. So the sample of interest, we have used this with cells as well as with tissues. The sample of interest is subjecting to increasing temperatures. And then quantitative mass spectrometry is used to quantify the amount of remaining soluble proteins at each of these temperatures. The readout is actually coaggregation curves of these proteins as the temperature increases. So how do we learn from this about protein interactions? Well, the principle is that proteins that interact with each other are stabilizing each other, and therefore they may have similar aggregation profile as the temperature increases. And this works actually quite beautifully, and you can capture or predict interaction on a global scale for thousands of proteins in one go in a sample. So Yutaka Hashimoto, at that time a postdoc in my lab, was the first to apply this method to viral infection, and he applied it to studying HCMV infection. And in this case, he took exactly this approach, but investigated samples at different time points of infection. And by looking at these co-aggregation profiles as the infection progressed and how these co-aggregation profiles change, we could predict which interactions are maintained during infection, which ones are formed only specifically at a certain time point of infection, or which ones are disrupted as the infection progresses. And more recently, we have further developed this approach, and Joshua Justice in my lab, a postdoctoral scientist, has applied it for studying HSV-1 infection. Tavis Reed, a Ph.D. student, now has developed machine learning algorithms and has further advanced this approach in order to be able to study different types of protein classes, like membrane-bound proteins and proteins that are harder to detect normally. But I want to kind of give you an idea of what we can find with this method. So we can, of course, monitor protein complexes that we all kind of know that they exist, functional protein complexes that have been reported, such as those that are in the Quorum database. But in the same time, you can actually uncover de novo interactions that were not previously known, either virus-host or host-host interactions. And I want to give you one example from one of our long-standing interests in my lab. We have worked in this field for over 15 years. And how we have gained new information by applying this thermal coaggregation profiling to the concept of nuclear DNA sensing. So upon infection with nuclear replicating viruses, such as HCMV or HSV-1, the viral DNA is surrounded by the viral capsid. It traffics along microtubules. The viral capsid docks at the nuclear periphery here at nuclear pore, and then the viral DNA is inserted inside the nucleus here. This is a moment in time that is very important, and these sort of dynamic events at the nuclear periphery that are important in host defense against viral infection and initiation of innate immune signaling. By recognition of this viral DNA by nuclear DNA sensors. And some years ago, we characterized this protein IFI16, interferon-inducible protein 16, that became the first known nuclear DNA sensor, so a host protein that is able to recognize viral DNA in the nuclei of infected cells and somehow recognize viral DNA from our own cell DNA in the same compartment. And here you can see IFI16 in green that quickly goes to the nuclear periphery upon the binding of the viral capsid at the nuclear periphery. So this was quite an interesting sort of time in this field of DNA sensing, but there are so many questions remaining. Like, we do not understand exactly how signaling happens downstream of this binding of IFI16 to viral DNA, or even what makes this protein go to the nuclear periphery to bind to IFI16, to bind to viral DNA. So from the thermal coaggregation profiling, we discovered that IFI16, this DNA sensor, very early in infection, 1 hour post-infection, interacts with a complex that contains the DNA damage response kinase, the DNA PK. And this interaction happens early in infection you can see this by these co-aggregation profiles, and then it's lost as the infection progresses. And we went on to further characterize this, and I show you just some of our findings in the interest of time. But we found that these proteins co-localize at the nuclear periphery at sites of viral genome deposition. And furthermore, we showed that IFI16 is needed for recruiting the DNA PK, so this DNA damage response kinase, to incoming viral genomes at the nuclear periphery. And this is the phosphorylated form of DNA PK, the active form that you see here. So if IFI16 recruits this kinase to incoming viral genomes, we asked whether IFI16, whether this can be a feedback loop, a regulatory feedback loop, and IFI16 may be a substrate of DNA PK. So we performed phospholipid enrichment analysis to study IFI16 phosphorylation status in a multitude of conditions, either with or without DNA PK inhibition, with and without DNA damage, inducing DNA damage by treatment with bleomycin, and with and without HSV1 infection at these very early time points of infection. And what we found is indeed that DNA PK can and does phosphorylate IFI16. We confirmed this also with in vitro assays. And it phosphorylates specifically this threonine 149 that is localized in these disordered regions between pyrene domain that is needed for oligomerization and the heme domains that bind to viral DNA. So IFI16 recruits DNA PK to the nuclear periphery, and DNA PK in turn phosphorylates IFI16. And that phosphorylation is actually required for IFI16 antiviral functions, because when we mutate that site, IFI16 is less able, or the cells are less able to induce interferon response and antiviral cytokine expression upon infection. And in agreement with this, if we inhibit DNA PK, we observe increased viral titers for HSV1. So what this technology allowed us is to find this elegant dance between DNA sensing, nuclear DNA sensing, and DNA damage response, in particular DNA PK in immune signaling, whereby IFI16 goes to the nuclear periphery, binds to viral DNA, recruits the kinase DNA PK, which in turn phosphorylates it, and that dynamic phosphorylation is important for IFI16 for inducing innate immune signaling. So this is one example of a dynamic event at the nuclear periphery that happens early in infection. But I told you earlier that actually the remodeling of the structure of the nuclear periphery is critical later in infection when the viral capsid has to egress from the cells. So the virus induces these infoldings through which the viral capsid exits the nucleus. So from the thermal coaggregation profiling, in addition to learning about protein interaction, you can actually learn about how infection changes the property of a given protein, either a viral protein or a host protein, and whether its aggregation profiles start to change as the infection progresses. So here is Lamin B in uninfected cells compared to different time points post-infection, and you see how its thermal profiles, thermal aggregation profiles, as the temperature increases, they change with infection. And this is in contrast with, let's say, if I select and show you a member of the nuclear pore complex, so another protein at the nuclear periphery that has fairly stable thermal aggregation profiles during infection. And furthermore, if we try to find out what this change in a profile means, it actually represents the disruption that I show you here, and also association with viral proteins that are critical in this capsid egress and virion stability. So for example, Lamin B1 here, late in infection, suddenly has same coaggregation profile with this viral protein PUL26 that plays a role in virion stability. So when we further integrated analysis of post-translation modification on top of this knowledge of interaction, we started to find out other regulatory mechanisms that are critical for driving or changing the structure of the nuclear periphery. And Laura Murray-Nerger, who was at the time a Ph.D. student in my lab, she discovered lysine acetylation on Lamin B1 that she showed when this Lamin B1 becomes acetylated at lysine 134, the integrity of the nucleus is restored, and the virus is no longer able to create these sort of infoldings. And this resulted in sequestration of the viral DNA inside the nucleus and viral capsids that remained inside the nucleus, and then reduced production of virus particles, both here intracellularly and extracellularly, as shown here. But more recently, Laura was able to show that what we stumbled upon by studying HCMV infection is actually even more broadly relevant. So first of all, she showed that also during infection with HSV1, so another nuclear replicating virus, an alpha herpes virus, again, the acetylation at the nuclear periphery of the Lamin B1 protects the integrity of the nuclear periphery and does not allow the viral capsid to egress from the nucleus. And then she showed that this is more broadly relevant in uninfected cells and that it's a mechanism that can control DNA damage repair choice and cell cycle progression. Specifically, she showed that when Lamin B1 is acetylated, this promotes intralaminar interactions. Therefore, the stiffness and rigidity of the lamina is stronger. But that leads the chromatin to be excluded from its interactions with the lamina, the interactions that are really critical for DNA damage repair. And then she showed that that leads to impaired recruitment of important DNA repair proteins to double-stranded breaks repair, such as 53BP1 in particularly. And that leads to a slower DNA repair and also persistent cell cycle activation of the G1 to S checkpoint and control of this DNA repair pathway choice in S phase, so for homologous recombination versus canonical non-homologous end joining. So it's an example of how by studying remodeling event during infection, we found a mechanism that is more broadly regulated and important during cell cycle progression and regulating the choice of DNA pathway, DNA repair pathway. Okay, so I showed you how we used interaction studies, either targeted or global, to learn about dynamic events that happen as the infection progresses. But we became quite more and more interested in understanding really structure-function relationships during viral infection. And in order to accomplish this, we turned some years ago to spatial proteomics. And I want to show you where we started and where we are going next. And I'll finish with some work that is not published and that will tell you about some of the new directions in the lab. So some years ago, we used spatial proteomics to start to understand what are the changes that happen in organelle composition and what are the viral proteins that may be targeting these different organelles in order to induce these changes. So in this case, we integrated live cell microscopy to find the specific time points when we observe those structural changes in organelles. Then we combined that with organelle fractionation, and we quantitative mass spectrometry, where we analyze the composition of these different organelle fractions. And then we use supervised machine learning to start to assign different host and viral proteins to different subcellular compartments as the infection progresses. And this was really eye-opening for us at the time. We found functional translocations of proteins from one subcellular compartment to another during infection. We found changes in the composition of organelles, and some that we further investigated in the future in our next studies. One such unexpected finding at the time was that proteins that are belonging to peroxisomes, so peroxisome proteins, increased in abundance during infection. And this was accompanied, we found out by using microscopy, by an increase in the overall number of peroxisomes during HCMV infection. And it was not only the number that was increased, but also the structure of peroxisomes that was altered during infection. So in uninfected cells, you can see here on the left-hand side, peroxisomes are spherical and uniform, quite pretty in size, in shape actually, and quite uniform. But late in infection, they become either fragmented, and the ones that survive, they are quite large, and we quantify this to show that they are elongated and curved. And by integrated further structural microscopy and mathematical modeling, we learn about how peroxisome biogenesis is regulated in mammalian cells. And then by using proteomics and lipidomics, we started to assign function to this. So specifically, we found that enveloped viruses, such as HCMV and HSV1, induce this increased number of peroxisomes, followed by changes in morphology. But this is not driven by infection with non-enveloped viruses, such as this adenovirus. So this is really nicely temporally regulated. So early in infection, there is first this growth and increased abundance of peroxisome proteins involved in the growth and fission of peroxisomes, so peroxisome biogenesis. While later in infection, there is an increase in protein abundance of peroxisomal proteins that are involved in synthesizing lipids called plasmalogens. So this change in structure that you see here, it activates the surface of the peroxisome and enzymes and proteins there, such as GNPAT, that are controlling the synthesis of plasmalogens, these lipids, that we then show to be important for the secondary envelopment. So they are integrated inside these virions, and they are critical for making infectious viral particles. And in agreement with this, we took cells from patients that have peroxisome disorders, and by infecting them with different envelope viruses, we found that they are quite resistant to viral infections. So these viruses need this metabolic function of peroxisomes for making lipids that are integrated in their virions. So this was unexpected at the time, and it was provided us with kind of a new outlook to peroxisome functions during infection, because for over 10 years, we knew that peroxisomes are really important in innate immune responses. They have MAVs, and they are involved in host defense responses early in infection. But now we found that these viruses have acquired mechanism late in infection to turn this peroxisome antiviral organelle into suddenly a proviral organelle by regulating lipid synthesis. So altogether, kind of these sort of studies have painted for us a picture of these dynamic structure functions relationships, and we wondered a few things, like one, can we start more and more to understand what is the function, what are the functions of these different structures? How are they driven and what are their functions? But also, are they coordinated events? Are these organelle remodeling events actually working in intracellular communication and even intercellular communication? And indeed, so this is not published, what I'll show you next, but indeed we know that organelles don't function as separate entities, but they rather form these dynamic intracellular networks. And this is accomplished by membrane contact site proteins that bring organelles, that tether organelles in close proximity, like 10 to 30 nanometers. And by doing this, it allows for transfer of biomolecules, such as lipids. It allows the recruitment of different proteins that also regulate the functions of these organelle contacts. And here is an example of one of our movies, where in cyan you see the ER, and in red you see mitochondria, and it shows this sort of dynamic interactions between these two organelles. So we wonder, how do viral infections change or regulate membrane contact site proteins during infection? And we predicted that many of the changes that we see in this organelle structure and function have to rely on the regulation of these membrane contact site proteins. But to our surprise, actually, MCSs have not been the focus of study during viral infection. And this is perhaps also driven by, in part, by the fact that these are not easy proteins to study. They are membrane-bound proteins. Many of them are low abundance proteins. There are no antibodies available for many of these to study. So this probably has driven some of this lack of knowledge in understanding this. Also, the method of choice, microscopy, for studying membrane contact sites and organelle contacts usually provides the opportunity to study one contact at a time or just a few. So we asked, however, can we actually understand how these organelle remodeling events are globally regulated and whether MCS proteins play roles in this during viral infection? And the work that I will tell you has been driven by this super talented Ph.D. student, Caitlin Cook, who worked together with Ileana and William in my lab. So in order to design an assay to monitor MCS proteins, mass spectrometry is in a way ideally suited for that. And the reason for this is that this is one of those cases where we know that protein abundance is directly connected to the extent of contact. So in other words, the level of the MCS is really critical for the amount of contact between organelles. So this is shown here. Let's say the knockdown of this protein, PTPRP51, this tether protein, when this protein is knocked down, you can see how that abolishes the interaction between the mitochondria and ER there. While when it's overexpressed, it enhances, it expands the interaction of the phases, the interaction between mitochondria and ER. So we therefore decided to design a targeted mass spectrometry approach that is based on parallel reaction monitoring to try to detect all these membrane contact site proteins in human cells. And this was quite a lengthy process. So Caitlin had to manually curate a library of functionally characterized MCS proteins. So we took into account their cell type specificity, we took into account tethers and interactors. And then she had to design a library of parameters that would allow us to specifically detect only those particular proteins by mass spectrometry. So she defined their detection parameters and unique peptides for their detection and built spectral library for all MCS proteins. This is way easier said than done, and actually it was quite an iterative process and a lengthy process. But once we had this, we could apply it now to measure the abundances of MCSs in one go simultaneously across different time points and conditions, and use that information to infer whether new contacts or change contacts between organelles are occurring. So our assay, so far, what it can do is simultaneously detect 90% of the known MCSs, and that contains all major organelle interfaces, including peroxisome lipid droplets, and all functions that are mediated by MCSs. So once we had this assay, we then applied it to the 4 viruses that I mentioned, these 4 envelope viruses that we study in my lab, the HCMV, HSV1, influenza A, and the beta coronavirus OC43. And let me walk you through this heat map. So what you see here, every column represents an MCS protein, and they are classified, or they are clustered according to the organelle contacts that they facilitate. So these, for example, are ER mitochondria, these are ER Golgi, and so on. And every row represents a different time point of infection. And what you immediately, hopefully, can see, the color represents changes in protein abundance during, compared to uninfected cells. So what you can immediately see is that HCMV infection triggers this global upregulation of in MCS protein abundances. And we have observed this after infection with the laboratory strain of HCMV, as well as a low passage strain that has more clinical relevance. This is quite in contrast with changes that happen during HSV1 infection, influenza, or OC43. So HSV1 triggers more subtle changes, but also more discrete changes. So they regulate certain MCS proteins in a specific manner. Influenza A downregulates many MCS protein abundances, as does OC43 infection. And of course, these viruses have replication cycles that are much shorter than the replication cycle of HCMV. So what can we learn from this? Well, first of all, just to kind of show you that what we find in a way really makes sense, I'm going to first continue on this theme of the peroxisome functions that I told you earlier. Something that was quite evident from our study was that the all protein tethers that are known to facilitate the contact between ER and peroxisomes, these 4 proteins listed here, they were all increased in abundance during infection with HCMV. And that made a lot of sense to us because it's known that that this ER peroxisome contact is critical for plasmalogen synthesis. So plasmalogen synthesis is initiated in the peroxisome, but then it has to be continued in the ER. And it's also important, this contact, for peroxisome growth. So we asked whether the increase that we see in protein contacts here, in their abundance, actually represent an increase in ER peroxisome contact, and that whether this happens during infection. And indeed, this was the case. So here in uninfected cells, you can see how these round and spherical peroxisome that I showed you earlier are quite dynamic, and they track along these ER tubules shown here in cyan. However, after infection, they become much more stably, they become enlarged, right? They're quite large peroxisomes. And they become much more stably anchored on the ER membranes, as shown here. And we were able to also perform 3D reconstruction and show how peroxisomes have increased contacts with ER membranes and that are stable in nature as the infection progresses. So what's the meaning of this, and what is the function, and does this tether actually, is the tethering important for the structural changes that I mentioned earlier with those curved peroxisomes or the increase in peroxisome numbers? So the tethering, the main two tethers here are ACBD5 on peroxisome side and VAP-B on the ER. And what we noticed is that, first of all, as expected, if we compare peroxisomes in uninfected and the infected cell, this is just for interest of time shown at a late time point of infection, we see that increase in peroxisome membrane area, and we see the increase in peroxisome numbers. When we knock down ACBD5, suddenly we find that peroxisome numbers are still increased, so somehow there is some sort of negative feedback loop that was not previously known. But the ability of these peroxisomes to become enlarged is lost during infection. And indeed, if we look at ACBD5 localization, we find that it localizes specifically only to enlarged peroxisomes during infection. If we do the opposite experiment and we overexpress this tether protein, ICBD5, we find that the number of peroxisome is decreased, but the size of peroxisome is increased. It forms this large peroxisome. So ACBD5 is required for these proviral changes to peroxisome sizes that I showed you earlier. So we also showed that this is indeed contributing to the viral titers changes that we reported earlier for HCMV. And we found that the same is true for influenza A and the beta coronavirus OC43. So these envelope viruses that seem to all need this temporal regulation of ER peroxisome contacts in order to control their functions. But I want to spend kind of the remaining few minutes that I have here on something new that we discovered that is quite intriguing, and it's based on these observations that we had on ER mitochondrial contacts. So to start by putting this in broader context, so we have been interested in the regulation of mitochondria for a while during infection, and it's very well known that many viruses are controlling mitochondrial fragmentation and fusion during infection in order to control apoptosis, immune signaling, and ATP production. And usually, increased fragmentation that is driven by many viruses is connected with reduced ATP production, so reduced bioenergetic output. However, HCMV infection kind of presents a conundrum, a paradox, because mitochondria is prominently fragmented after the infection with HCMV. However, somehow, they are also bioenergetically enhanced. And the mechanisms that underlie this have remained kind of mysterious. And something that we noticed from our MCS-PRM mass spectrometry analysis is that proteins involved in ER mitochondrial contacts are increased as the infection progresses. And this in particular happens at 48 hours post-infection, which is the time when we see that pronounced mitochondrial fragmentation happening, but also when we see many cellular changes in cellular metabolism, increased flux through glycolysis, increased oxidative phosphorylation. So we wonder whether these changes in ER mitochondrial contacts are actually somehow driving this increased, this interesting phenotype that we can't explain. But first, in order to look at this, we had to convince ourselves that changes that we see here in protein contact, in protein abundances, actually reflects a change in organelle contacts. So using fluorescent microscopy and live cell microscopy, we monitored the interaction between mitochondria and ER. And in uninfected cells, we noticed this really traditional interaction that has been reported many times before, in which these ER tubules are weaving and crossing across the mitochondria. And you can see these little constriction sites where on mitochondria, where the ER membrane, ER tubules are crossing. And this is a dynamic interaction, as you see here. However, after infection, the interaction changes quite strikingly. The mitochondria become encapsulated. They become much more stable in their interaction with the ER membranes. And when we also labeled the outer mitochondrial membrane, we could confirm that this contact between mitochondria and the ER is increased, but also that this is asymmetric, that it happens on one side of the mitochondria where the contact is increased. So this is quite different. This sort of encapsulation and stable interaction is quite different than the dynamic ER tubule crossings that are usually reported for these interactions, and presents a new mitochondria-ER contact structure that has not been reported before. So we use live cell super resolution microscopy. We use SORA to gain a better understanding of what we are looking at. And indeed, we found that the ER encapsulates the mitochondria like this in little pockets. And this is a stable and asymmetric membrane encapsulation. So we term this structure MENCs for mitochondria-ER encapsulations. And we monitor the formation of MENCs during the progression of infection. And we noted that as the infection progresses, this contact, the MENCs, become the primary contact type for ER mitochondria. And we also noticed that as these MENCs are formed, this have increased in this stable type of interaction between these two organelles, and the dynamic interaction previously noticed is decreasing. So we therefore ask, what are actually the ER mitochondrial MCS proteins that are localizing to these MENCs, and what is downstream of this structure formation? So in looking at our mass spectrometry data, the two proteins that were the most abundant at this ER mitochondrial contact were this tethering complex that is formed by PTP-IP51 and VAP-B. And this complex is known to be involved in calcium flux, in regulating bioenergetics and autophagy. So we monitor the localization and the temporality of those localizations for these two tethering partners as the infection progresses. And that's allowed us to discover the temporality of this. And we found that first MENCs are formed, after which VAP-B is recruited at around 48 hours post-infection, and then PTP-IP51 is recruited at 72 hours post-infection. And both of these proteins, VAP-B and PTP-IP51, gain this asymmetric localization on one side of the mitochondria. And we further validated this interaction using proximity ligation assay to confirm the temporality of this interaction, and also observed the interaction for the endogenous proteins. And we could see how late in infection this actually happens. So since PTP-IP51 was the last one recruited to this, so MENCs formed, then VAP-B, then PTP-IP51, we asked what's the function of this. And to address this, we used another targeted mass spectrometry assay that we developed recently in the lab. And this was developed by Michelle Kennedy, a really talented Ph.D. student in the lab. As I told you earlier, these viruses, herpes viruses, are really large viruses. They have large genomes. We lack antibodies for most of these viral proteins. So detecting them and quantifying them accurately during infection is not trivial. So Michelle has developed this targeted mass spectrometry assay to detect the majority of the proteomes of these viruses, HCMV, HSV1, and KSHV. And she showed that she can capture the temporal expression of viral gene expression. She can monitor the temporal effects of drugs or small molecules or genetic perturbations. And together with Tom Cristea, we also showed that we can apply this not only in cell culture, but also to animal models of infection. So therefore, we used our HCMV version of this assay, but this time to our condition of PTP-IP51 knockdown to find out what's the functional consequence of this recruitment of PTP-IP51. And what we found, so here is the control knockdown, and this is the PTP-IP51 knockdown. And you're looking at a few viral proteins illustrated here. But what we found is that the knockdown of PTP-IP51 does not impact the expression of immediate early viral genes, but rather it decreases the levels of delayed early and late viral proteins, so proteins that are synthesized at around 48 and 72 hours post-infection. And this decrease in viral protein levels indeed leads to an over 100-fold decrease in virus titer, and this impact on the virus titer is maintained over time. So altogether, what we now understand is that this protein, this tether protein, PTP-IP51, increases in abundance during infection. It's accumulating asymmetrically at MENCs, and it's tethered to VAP-B late in infection, and this is supporting virus production. So I just told you, right, that late in infection, PTP-IP51, it's pro-viral, it's supporting viral production. But it turns out that the temporality is absolutely key, and this was something really surprising at the time, and then made a lot of sense to us. So if we prematurely increase ER mitochondrial contact, it turns out that this is antiviral. It's not proviral. If we set this interaction too early, it's not doing what we thought it would do. So for example, if we express a synthetic tether that brings ER and mitochondria together early in infection, we suddenly see a concentration-dependent decrease in virus production. So few questions. First of all, why is it proviral only late in infection, only after 48 hours, when this contact, if the formation of this contact is only proviral late in infection? And we think that it's because it needs the right context. It needs the mitochondrial fragmentation to occur, and it needs the localization of viral factors to the mitochondria. So from that spatial proteomics study that I mentioned earlier, I told you that we found out which viral proteins localize to different organelles in order to perhaps induce some of these changes. And we found, and this is the work of Cora Betzinger in my lab. She just published this last year. But we found that this previously uncharacterized viral protein localizes to the mitochondria. And by using live cell metabolic assays, seahorse assay, she showed that this viral protein is necessary and sufficient to increase oxidative phosphorylation. And we think that this is actually downstream of that MENC formation when UL13 now is localizing there. She asked, how is UL13 accomplishing this? And to do this, she analyzed its temporal interaction, protein interactions, as the infection progresses. And she used a program, a computation platform that we developed a few years ago called InterVista. So she infected cells with this virus that had UL13 tagged with green fluorescent protein. She performed immunoaffinity purification at different time points of infection and used this computation platform to visualize these interactions. And she found that at 48 hours post-infection, the time when the mitochondria is fragmented, when we see MENC formation, UL13 goes to the mitochondria and interacts with Cristea remodeling complexes. And also using super resolution microscopy, she showed that by itself, UL13 is sufficient to change the architecture of the Cristea, to remodel the Cristea structure, the surface and the volume of these. So this represents the first viral protein that can modulate, that can localize to the mitochondria and modulate the ultrastructure of mitochondria by itself in order to increase bioenergetics. So I told you that this proviral function happens late in infection, and why we think that happens. But lastly, if we come back to this idea that if we tether this earlier, suddenly this becomes antiviral, the question is, why does that happen? Well, and using this synthetic tether, we found that if ER and mitochondria are forced to interact too early and maintained as a stable interaction too early, it actually activates STING. And thereby, it activates the TBK1 and IRF3 signaling cascade and induces cytokine expression. So in agreement with the activation of this broad host defense mechanism, we see that a tether, forcing this interaction early on, also decreases the virus titers for HSV1 and influenza A in a concentration-dependent manner for this tether. And this is perhaps why, for these viruses that have much shorter replication cycles, we see that early in infection, they immediately start to downregulate these ER mitochondrial contact proteins that otherwise, if they would force this interaction to happen, would induce immune signaling. Okay, so with that, what I told you is how these ER mitochondrial contacts are temporally restructured and how the temporality of the MCS regulation is critical, and how we have discovered this new structure, a mitochondria-ER encapsulation, that we demonstrated that it's necessary for virus production and it's pro-viral. And how this rewiring of membrane contact site is much broader. It happens quite globally during HCMV infection. And we developed this assay to be able to monitor all these MCSs in the same time. And how this allowed us to find virus-specific as well as shared MCS rewiring events, such as the ER peroxisome contacts. So with that, I'm going to thank the people involved, the ones, in particular, the ones that I highlighted some of their work today, my collaborators, the funding, and my phenomenal lab. And I want to thank you for your attention, and again, the honor for inviting me here today. And I'm, of course, happy to take questions.
Okay, thank you. Anyone's free to use the mics in the aisles, please do. There's a couple questions online. I'll do my best to read for you. This came in early in your lecture. So
sure.
Backwards here. Basically, the temperature, this is from someone here, Lumen Zhang. Basically, the temperature is changed with air consumption, especially high temperature. Did you change the culture condition, especially oxygen and CO2 consumption?
So, okay, so this probably refers to the thermal coaggregation profiling, where the principle is that you're inducing this aggregation of the protein because you're increasing the temperature. Of course, that at that time, you're just simply aggregating the protein, so you're not trying to preserve the cell functions in a cell. It's mainly to aggregate the protein and see which what aggregation profiles and are similar to each other in order to predict interactions. But something that that's a very nice question, because something that we have actually done is now to revisit a few things for that workflow. In particular, the temperatures that are actually needed in order to get those curves. And we find that you do not need to go that high in temperature in order to be in the sweet spot where you best predict coaggregation profiles. And also, many proteins, as you probably guess, this is computationally intensive, right? So, and many proteins do not behave very well, and they are having curves in all different directions, and aggregating proteins or membrane-bound proteins do not behave very well. So Tavis Reed in my lab has optimized now lysis buffer conditions to try to capture more and more of the proteome. But he also has tested experimentally and also computationally monitored what are actually the temperatures. So we need only 5 temperatures in order to get really great information about aggregation curves. And this brings it to a different sort of ball game in terms of multiplexing this and making it much faster. Yeah. But, and then he also developed, as I said, these machine learning algorithms for better analyzing the data. Yes?
Yeah, I have 2 questions. One is to the Lamin acetylation. Oh, yeah. Do you know the enzyme that does it? Is it a specific acetylase or?
So we have only very little dabbled in that for the moment. So I can tell you what we know so far. We know that it seems like P300 is the acetyltransferase that does that. For the deacetylase, we tested it with hijack inhibitors, and we didn't see a difference. We tested, therefore we think it's sirtuins. We started to look at sirtuins 1 and 6, and we noticed that sirtuin 6 is going to the nuclear periphery, and therefore is kind of at the right time and place to try to deacetylate that. But we have not yet looked into that to validate it.
Yeah. Because this could be manipulated to, therapeutically potentially.
Yes, exactly. Yes, yes. And as we all know, these viruses are arresting the cell cycle, right? So they replicated this G1S sort of checkpoint in that, so it's perfect for the host replication machinery to be expressed, but not for host replication to happen, right? So it makes sense that acetylation, as we see it in uninfected cells, is increased exactly then.
Yeah, and the second question is to the tethering, the last topic. Do you think it's a more general phenomenon, or it can happen in all infections or even whatever perturbations the cells are at? And so, if so, because it can, I was thinking that it can affect other kinds of experiments. For example, cellular localization experiments can be changed because of that, and not because the protein actually changed the localization. They can look like their localization.
So I think it makes so much sense, and I'll come back to that particular tethering and tell you why I think it may or may not be. But it makes so much sense for membrane contact site proteins to be regulated during infection, because if we think about it, this is such a so critical proteins for intracellular communication, for this very fast signaling that we see and responses. So when we look at all those structural changes, it's impossible for a structural change like that not to immediately influence its environment and also communicate that. And what better way than even direct contacts between organelles that can quickly move small molecules or recruit proteins. I've been for a while fascinated with also the concept of mechanoresponses and mechanosensing that can happen during infection, right? So I think that those are clearly involved here as well. Now, coming back to that particular tether, I think in terms of the, let's say, depending on which, so for the MENCs, for these structures, I actually think that this is probably fairly specific. So if we just induce mitochondrial fragmentation, or if we even perturb the ER functions, we do not see MENCs forming. So we think that this is actually having to do with that really interesting phenotypes of mitochondria being fragmented, but being still bioenergetically enhanced. So we now show and have preliminary data that we think that this is protecting the mitochondria, and perhaps decreasing mitophagy, for example, and among others. So this is the work of Will Hofstadter here and Caitlin Cook. So I do not think that it's going to be relevant to all viral infections because of this. However, we now have evidence that it's relevant in other circumstances. We think that it's relevant in certain neurodegenerative diseases, but we also think that it's relevant in cancer. So we looked at some cancer cells because they trigger a similar sort of change in cellular metabolism like HCMV, right? So we now see that MENCs do form there. Right?
Yeah, so worth remembering as a phenomenon. Yeah, thanks for this inspiring talk.
Okay, I have a couple more. So this is from Dr. Roland Owens, also at the NIH. Might be related to that. Could the virus DNA binding protein be recognizing DNA ends without telomeres? Do the same proteins recognize circular viral DNA?
Yes. So that's really interesting, and I don't know. So this is probably referring to the DNA sensing story, the nuclear DNA sensing, and how how this how this host protein IFI16 recognizes viral DNA instead of host DNA. And one thing that I can say, we have done this, but this was done in vitro, where we looked at circular DNA versus linear DNA. At the time, we were speculating that it's going to bind more to the linear DNA because maybe it's going to find this as damaged DNA. But we find that it binds to both circular and linear in a similar fashion. We have since then done now ChIP-seq and ATAC-seq experiments where we show that it binds to viral DNA in a sequence-independent manner. And this is, it's known already that it's, so the pyrene domain has all these basic residues, and we found certain residues that can be mutated, and we found how those residues are involved also in the oligomerization of the protein after binding to viral DNA. Yeah, there are so many things to say there, so you can stop me when you want.
Okay, well, I have another one. Maybe we can sneak in one more before we close. This is from Ron Germain at the NIH here. You showed that MENC form early before the adhesive interactions with the proteins that contribute to virus replication. What allows formation of MENC that involves membrane-to-membrane interaction of the organelles, but not the molecular pair you mentioned as critical for ER mitocontact?
So can you please repeat that, because I missed the beginning. So it's about the MENCs, right?
MENC,
Yes.
is that actually
MENCs.
pronounceable?
Oh, it's fine. Yes.
Yes, okay,
Chris,
so you
sorry,
showed
you
that MENC form early before the adhesive interaction with the protein that contributes to virus
So yes,
replication.
because so the ER mitochondrial contact actually is formed by many proteins. So remember that heat map, and I showed you how many of them are known to form that ER mitochondrial contact. But then these proteins, the VAP-B, I think that's what the question refers to, and PTP-IP51 are recruited there. In fact, if you look at PTP-IP51, it's usually distributed, diffusely localized, and suddenly you see how it relocalizes and it goes to this tethering with VAP-B, and how it's asymmetrically localized on the mitochondria. So, and that's known, I mean, that's not known for infection, or not known that it happens in an asymmetric thing, but that relocalization is known to be important for the formation of this tethering complex. But there are other proteins that earlier on are bringing these two organelles into contact, and that are increasing in abundance, as I showed there.
Maybe if you want to close this out.
Thank you for all those great questions.
Yes. Thank you very much, Ileana, for this great talk. I'm sure it will be inspiring for a lot of people. And thank you very much for coming, and wonderful attendance in person and online.
Thank you so much. Thank you again.